One of the most important evolutionary developments that occurs over time in the animal kingdom is an animal's ability to fight or run from predators. Geckos and many other types of lizards have developed nifty adaptations in which they are capable of dropping off their tails when they believe a predator has grabbed them. This distracts the predator for long enough for the lizard to escape. This is not a one-time thing that they can do either. Geckos are then able to regenerate their lost tails, which include a large portion of their spinal cord, in just 30 days. This is faster than any other lizard. This is a great mechanism to help geckos and other lizards escape from predators. However, as you might imagine, regenerating an entire appendage is an incredibly energy-intensive process. Once the special connective tissue in the gecko's tail breaks off, a sudden change occurs in the small portion of the tail that is left over. This change occurs as a result of hundreds of genes being turned on in response to the loss of the tail. The cells then almost immediately begin to proliferate while secreting proteins which are known for supporting stem cell growth. This results in the beginnings of new tissue being formed in a new spinal cord, which take the place and function of the tissue that had been dropped off with the tail.
So what are stem cells, and how could geckos use them to grow back entire limbs? Stem cells have the remarkable ability to develop into many different cell types in the body during early life and growth. They divide essentially without limit, and each time a stem cell divides, it has the potential either to remain a stem cell or become another type of cell with a more specialized function. They differ from other cell types in two important ways. The first is that they are unspecialized and are able to renew themselves through cell division, even after being inactive for long periods of time. The second is that under certain conditions, they can be induced to become tissue or organ-specific cells with special functions. A very important type of stem cells are neural stem cells, which are able to self-renew and proliferate without limit to produce progeny cells that terminally differentiate into neurons, astrocytes, and oligodendrocytes. Neurons are responsible for transmitting information to other neurons, muscle cells, or gland cells through action potentials or neurotransmitters. Astrocytes and oligodendrocytes are known as glial cells, which support functions for the neurons. Neurons and glial cells make up the nervous system, and most can be found in the brain and spinal cord of animals. Recently, research done by Gilbert and Vicarius proved that neural stem cells/progenitor cells, or NSPCs, help geckos regrow entire tails. This makes sense because the replacement tail includes a regenerated spinal cord with a simple morphology. They proved this by monitoring a protein called SOX2.
In this figure, one can see the 7 distinct morphological stages of leopard gecko tail regeneration. These stages can be grouped into 3 different periods. The first 2 stages comprise wound healing, the next 2 comprise early regenerative growth, and the final 3 involve late regenerative growth. In stages 1 and 2, one can see the spinal cord, marked SC, retracting slightly into the body. By stage 3, a new arrangement of epidermal layer cells is evident, marking the first growth of the regenerative spinal cord. This is marked RSC. According to the study, these epidermal layer cells express SOX2, a protein essential to neural stem and progenitor cells, all throughout tail regeneration. The figure makes this clear, as one can see all new tail growth originating around these cells.
From a biomedical perspective, the applications of this research regarding regenerative capabilities in leopard geckos are immense in nature and still working to be completely understood with respect to human capacities. The article by Gilbert and Vicarius in 2017 noted the utilization of the SOX2 signaling moderator, which in stem cells is associated with maintenance of stem cell proliferation, apoptosis, invasive properties, cellular regeneration, and cancerous tumorigenesis, as SOX2 participates in autocrine, paracrine, and endocrine signaling pathways as an intracellular moderator of gene expression and transcription capabilities. The genes transcribed due to SOX activation or inhibition produce certain signals which are interpreted by the cell itself, adjacent cells, or distant cells via signaling pathways. There is a lot of hope for this type of research with regard to regenerative medicine, particularly in human spinal cord injuries. Humans generate a lot of scar tissue during spinal cord injury, resulting in the belief that humans cannot regenerate spinal cord tissue. However, with potential research from stem cells, this could potentially be a treatment in the future.